Hello, we're going to be taking a look at models of membrane structure. So you probably heard of the plasma membrane or the cell membrane, and what we're going to do here in this video is look at an old, outdated model and see what kind of evidence surfaced to actually falsify that model and allow us to accept the newer model, which is called the Singer-Nicolson model, more famously known as the fluid mosaic model. So it gives us a little insight into the nature of science and how basically the nature of science is such that when new evidence comes out, you can completely remove any of the previous ideas that were there. So first, a little bit about the Davison-Danieli model. If you just look here at this little diagram here, this kind of summarizes it. They thought that the cell membrane kind of looked like this, like a double layer, a bilayer of phospholipids, and that there was a layer of protein on either side of this thing. So here we have bilayer of phospholipids in the center, surrounded by a layer of protein either side. The reasons they thought this might be what the plasma membrane actually looks like is when they analyzed the contents of the cell membrane, well, they did detect the presence of phospholipids and they detected the presence of protein. So it's about figuring out the actual arrangement, and this was one proposed arrangement. Seems to make sense. Danieli and Davison came up with this idea. Evidence existed when they analyzed RBCs, red blood cells, that there were more phospholipids in there than were required to actually surround a red blood cell all the way. So that suggested that maybe the red blood cells are circled twice, and so that's why they came up with this double layer, this bilayer aspect right here. So that seemed to fit as well too. And they also knew that membranes, the purpose of cell membranes, they could act as a selectively permeable membrane, which means certain things could be blocked and certain things could be allowed through. It was also known at the time that proteins had the properties that would allow them to have this kind of filtering mechanism. So there you go, bilayer. The number of phospholipids seems to make sense from what they studied in red blood cells, and a layer of protein acting as a barrier or filter, giving it its selectively permeable properties. And this model. Here's another diagram of the same thing, and little electron micrograph pictures of this kind of showed it looked like there was a darker layer on the outside. It was known that proteins tend to show up darker in these images, so it looked like there were two layers of protein on either side of this actual cell membrane. So looks great. Davison-Danieli, fantastic work, great job, until new evidence starts to surface. I'm going to go through that evidence in a second, but here's the updated model. It's called the Singer-Nicolson model, also called the fluid mosaic model. This is the one that you studied. If you've never heard of this one before, it's because most textbooks don't talk about this model, the Davison-Danieli model, unless we're talking about showing how the nature of science works throughout. So what you know and what you should be able to sketch these days is this diagram of the plasma membrane, and you can draw your phospholipids there, and you know that there are proteins that are kind of interspersed throughout. Some of them go all the way through the membrane, some of them are only at the side. They're called peripheral proteins. Some of them are up here. There's like 5, 6, 7 different functions that these could be, and that'll be in another video, or I'm sure maybe you studied that a little bit already. And there's some cholesterol in the membrane as well that affects its fluidity. Now we think that these little guys kind of from the bird's eye view kind of swim around each other and kind of are moving around. Imagine like looking at a bunch of people floating in a swimming pool viewed from the top with different colored swim caps. You would see all these little colored balls moving around, floating on top. So that's the idea. Let's look at the evidence for that as well too. So the evidence for the fluid mosaic model comes in a couple of different forms. This kind of makes me feel gross because there's something, I don't know, is this a product in Japan? Maybe it's common in other places. I tried it before. It's quite nasty. It's called a Biore nose pack or skin pack. I'm revealing my skin properties to you. But basically, you take this thing, you wet your nose, and you place a kind of a little Band-Aid thing across your nose. And the point is, it's supposed to clear your pores. You let it sit there and it dries. And then when you peel it off, you literally get exactly what this looks like. So you see the surface, and then sticking out on the paper are these little bumps. And they're actually little bits of oil and grease, solidified oil from your pores, and the whole point of doing it is pouring this out, and you're left with all these little sad holes in your forehead or in your face. I don't know, it's meant to be a face cleansing mechanism type thing. Anyways, I stopped doing that because it was freaking me out when I was looking at it, and then it all came back to me when I had to watch and learn about this kind of stuff all over again. So anyways, what they did was they actually took the plasma membrane and used some really amazing techniques called freeze fracturing, and they were able to crack the plasma membrane right in the middle. And when they were able to do that and take a look inside, they actually noticed that there's these little bumps, kind of like bits of oil from my nose holes, that's gross, that were showing up there, which suggested that there were some proteins that were actually found inside the membrane, as opposed to what we saw in the previous model for the Davison-Danieli one, where there were 2 layers of protein on the outside. Now we have evidence that there's proteins actually embedded inside the layer, like stuck inside the layer, as you would imagine if that were the case with this. They didn't see this beautiful image right here, otherwise they would have freaked out. But this is what they actually saw, and they were able to determine that it means there's probably some proteins that could possibly be within the layer. Another cool thing they did was that they actually tagged proteins in the membrane with different fluorescent colors. And I guess what you can, here's a interesting way to think of it. So they have one cell here, and the proteins in this cell in the membrane are tagged with very specific fluorescent markers. They have another cell with different fluorescent markers, and they fuse this cell, and when you fuse them from 2 sides, and it just comes together, you would expect that those fluorescent markers would stay where they were. But after a time, the colors started mixing, which suggested that they were moving around, kind of like that model we said in the swimming pool. If you're looking from the top, you see some swim caps here, and then they start to actually move around. So these swim caps start getting mixed up. So after a while, this color actually started mixing and fusing, which suggested that there was some fluidity and movement in the actual membrane, hence giving us this evidence for the fluid mosaic model. The word mosaic comes from looking at patterns of proteins kind of spread out like a mosaic painting or piece of artwork, and it's all spread out, mosaic tile pattern like, I guess. So this shows the fluidity of the membrane. Phospholipids are moving around each other, and they're in the act of doing so, the proteins are being pushed around as well too. Finally, one last piece of evidence is it was figured out or known that some of these proteins, these membrane proteins, had different properties depending on the location of some of these guys. So some parts of them liked water or were hydrophilic, and some parts of them were actually hydrophobic and didn't like water. And that seems to make sense because based on the properties of phospholipids, we know that tails are actually hydrophobic. That's why they face each other to get away from the water out here and the water inside the cell over here. So it makes sense, I guess, that some of these proteins, if they're going to live inside the membrane, then some parts of them have to like water and some parts of them have to not like water. So the parts that are sitting here touching the tails are actually hydrophobic, and the parts that are actually touching the outside or the surface are actually hydrophilic, because there's a lot of H2O out here and a lot of H2O inside here. And it turns out, now that we understand the properties of proteins. Proteins are made up of amino acids. Amino acids, there are polar amino acids and non-polar amino acids. If you actually break down the structure of some of these proteins and study them, you can see that the lining out here, or even inside for the channel when we need to get things like glucose through, there's a lot of polar amino acids in here and a lot of non-polar amino acids over here for the parts that are actually touching the membrane. Pretty clever stuff, and a really smart way to analyze the evidence to get us from the Davison-Danieli model to the Singer-Nicholson model. Alrighty, there you go.